Okay, and good morning, good afternoon, and good evening, and welcome to the International Society of Biopharmaceutical Statistics, ISBS webinar series. Our committee includes Cindy Lu from Astrazeneca, Dongxi from Gilead, Henzhou from Merck, Rui Ling from MD Anderson Cancer Center, and myself, Haitao Pan from Senzhou Children's Research Hospital. We invited esteemed speakers from industrial, regulatory bodies, and academia to share their insights. And today we are very pleased to welcome Professor Sofía Villar and Dr. Mia Tackney from the University of Cambridge's Medical Research Council, MRC, a biostatistic unit. We know it is one of the largest biostatistic groups in Europe. Professor Sofía Villar is currently an MRC investigator and a program leader specialized in innovative clinical trial design. Her research emphasizes patient-centric trials, and she is a well-known scholar in adaptive randomization method. And Dr. Villar has extensive experiences in adaptive trial design and has led the design of many UK clinical trials. And Dr. Mia Tackney is a research associate at the MRC Biostatistic Unit, where she collaborates with Dr. Villar, and her research focuses on statistical methods for digital outcome measures in clinical trials. Without further ado, I will pass the mic to our speaker.
Thank you, Haitao, and thank you, Cindy. I will wait until our slides appear. Thank you for the invitation. It's really our great pleasure to be presenting our paper in this ISBS webinar. It will be a presentation that it would be two people. It would be me at one part and then Mia at some point, and then me again and then Mia. So the content of this webinar is based on this paper we just published that is called 8 Methodological Questions for Digital Outcome Measures. And I do want to start a little bit telling the background. Let's see if I can control the slides.
I'm not able to find, sorry. I'm going to share again the screen.
That's fine. We'll get it going. And meanwhile, I could check if everyone can hear me okay and Mia okay. So if not, please just let us know.
Okay, I passed the control to you.
So. Okay, let's see if it will work now. Okay, I think so. So I wanted to start the presentation by telling a little bit of the background on how this paper happened and came to exist. So a bit of how we get here. So Mia in 2021-2023 was doing a postdoc in the London School of Hygiene and Tropical Medicine and working on methods for missing data in trials that were using accelerometer data. And this was mostly for final analysis of these trials. In October 2024, Mia came to work at the Biostatistics Unit with me on a fellowship track position, and we started to think together what we wanted to work on as part of her fellowship application. And so we started looking closely at the 2019-2023 SV95C approval. I will describe this a little bit more. And we thought that combining the knowledge of missing data and design of clinical trials was something we could think of. So these are the papers that you can check out. Mia worked on with Fizz Williamson and James Carpenter, and this was part of our starting point. But where we started was looking quite closely at this approval of SV95 for Duchenne. So I'll tell you a little bit about what that is to make the context. So in 2019, EMA, the European Medicine Agencies, qualified stride velocity 95 centile, this is what SV95C stands for, as a secondary endpoint in patients over 5 years old for Duchenne. So Duchenne muscular dystrophy is a muscle wasting disease that affects nearly 70 boys born each year in the UK. These boys do not produce an essential muscle protein called dystrophin, and they become wheelchair users by their teenage years. And they unfortunately die young in early adulthood. There is currently not a cure for Duchenne, but there are several treatments being studied or tested in clinical trials. So this was a breakthrough for this specific community in 2019 when this measure based on an ankle-worn device that you can see in the picture was approved as a secondary endpoint. And actually in 2023, EMA qualified this measure as a primary endpoint, not a secondary endpoint, in patients over 4 years old. And this was the first ever qualification by a regulatory agency of a digital endpoint for use as a primary outcome in an indication. So SV95C is just a digital clinical outcome assessment that is measured by a watch-like device, but it's worn in the ankles rather than in the wrist, and during the patient's everyday life, and is passively tracking gait metrics. So specifically, SV95 will represent the speed of the top 5% fastest steps taken by, in this case, a child, during the measurement period. So this caught our attention immediately, and we thought this had a huge potential to think about it a bit more. And so after looking closely at this, which was a 10-year process, by the way, a regulatory process, we went into the guidance and say, what's the guidance around similar clinical outcome assessment measures that might be aiming or aspiring to do this similar thing? And we went to the Digital Health Technologies for Remote Data and Acquisition in Clinical Trials guidance. And we made a comment on it because we thought it was quite thorough in the technical aspects, but we were a little bit concerned that some of the statistical issues that we thought might be very important were a little bit not so clearly mentioned or addressed. And you can read that if you use the QR code, our comment in there. So after that, we went on in November 2023 to try to get people together in a knowledge exchange event, because we quickly realized that the process of approving an endpoint like SV95 involves multiple perspectives, and we wanted to understand how we could use those perspectives to shape our statistical methods priorities. So we created, we put people together, regulatory, industry, patients, and different aspects, even more methodologies, and we tried to discuss the challenges and the possibilities. And if you're interested, you can read, we published a paper in Clinical Trials that can give you ideas of the challenges around organizing multi-stakeholder events and how we went about that, but also the insights that we got from putting all these people together, which were many. And after that, with all of this in mind, Mia mostly, who is the driver of this work, and I wrote this paper with James around what we felt were the methodological questions that needed the most attention so that we could really unleash the full potential of digital outcome measures to deliver all the promise that we think they hold. And this is what we're going to talk about in the next part of the seminar. So at this point, I'll hand over to you, Mia.
Great, thank you, Sofía. So the 8 questions that we're going to ask about digital endpoints, digital outcome measures, they span different parts of this process. So some of the questions are going to be about the validation of digital outcome measures, and some of the questions are going to be about how you actually use them in trials. So we'll be talking a little bit about early phase trials and a little bit more about late phase trials. Now, when it comes to validating a digital outcome measure, something we have to keep in mind is that the data just looks very different compared to the data you get from in-clinic assessments. So first of all, you just have quite a lot of data. What I'm showing here is what accelerometer data looks like for just about 30 minutes. They take measurements 100 times per second in 3 dimensions. So it gets very heavy very quickly. And you can also take data from a much longer period. So here we're showing what accelerometer data from 1 week looks like. And here we summarize it into epochs. So here an epoch is 1 minute. Further, there's another step of aggregation typically. And in this example for Duchenne muscular dystrophy, they took stride velocity 95th centile as the summary of all of this data. And they showed that this is just as good, actually even better than the 6-minute walk test. But what do we really mean by saying that it's just as good or better than another outcome measure? Well, this is the process of validation. And quite recently, the Digital Medicine Society has come up with a framework called the V3+ framework to describe what that validation process is. And it breaks it into these different component parts. So the first component is called verification, and this is about the technical validation of the device, of the hardware, that's done on benchtop tests in the lab. There's also usability validation, which is about whether the device is acceptable, easy to use. Then there's these two components that have quite a lot of statistical aspects. So analytical validation, this is about the physiological metric. So when you're trying to measure strides, like you're trying to do for stride velocity 95th centile, you want to make sure that you can do this accurately. So that means that the strides that you measure and quantify with the device should be just as accurate as what you can do with the motion capture room. You also want to make sure that your measurements are repeatable. So if you measure stride velocity 95th centile in one block of time, and then you ask participants to do this again in another block of time, those values should be quite similar. You also want to have an idea about the robustness of this approach. So you may want to understand what's the impact of the measurement period. For stride velocity 95th centile, this was 180 hours. And you may want to understand the impact of contextual factors, such as if it's a weekend or a weekday. Then we come to clinical validation, and this is about being able to understand a clinical state. So you want to be able to know that you can discriminate between people with and without the disease. In this plot, you're seeing that those with Duchenne muscular dystrophy, shown in red, have a lower stride velocity 95th centile compared to their age-matched controls, shown in blue. And you can see by the gradient that there is a high correlation with the traditional outcome, the 6-minute walk test. So this is also showing convergent validity. You also want to show that the new digital outcome measure is sensitive to disease progression or to treatment. And if it is more sensitive than the traditional outcome, this is where it's possible to have sample sizes of trials that are lower, thanks to the digital outcome measure. Now, in this process of validation, there's quite a lot of methodological questions that we want to pose. So the first question is, what is the appropriate outcome granularity? As we saw, you can aggregate this data in many different levels. And some aspects of the validation, for example, the part about checking that the strides are accurate, this is on a different granularity compared to a lot of the other aspects, which is about the summary measure about stride velocity 95th centile, that large summary metric. So when you move between these different levels, there's lots of decisions being made about how to define outliers, for example, what to do with missing data. And there is not very much guidance on how those decisions should be made. So that's our first question. Our second question is, what is the appropriate measurement period? So for stride velocity 95th centile, they chose 180 hours as an appropriate measurement period. They considered factors like the variability of this outcome as the measurement period changes, but also things like burden, compliance, and battery life. But how to make this decision in other contexts is really quite complex. And another complex decision is how to choose the ideal summary metric. So in the example of Duchenne muscular dystrophy, they chose the 95th centile amongst other candidate metrics, such as the median stride velocity or the average distance walked. And they chose this because it was more sensitive to change than some of the other metrics. But in other contexts, it's not clear how you should pick this one summary that you're going to go for as your digital outcome measure. And finally, we know that when you're asking participants to wear a device in their daily conditions, typically unsupervised, this can lead to missing data. And because of the type of data we're looking at, it's quite difficult to even define what missing data is. You can see here that we've got days that are missing, but also parts of days that are missing. And very likely will be missing not at random, for example, if participants remove the device when they're less active. So how to define missing data, how to handle it, this is a very large question. So we've talked a little bit about the validation. I'm now going to hand over to Sofía to talk about the early phase trials.
Thank you, Mia. Hopefully I can move to the next. So the next question we posed within early phase, we just had one question, but this question encapsulates possibly many questions. But the main question in early phase is, one of the things that comes up from this high frequency data that is given by the digital outcome measure is the possibility of exploiting the granularity, this higher granularity of the measure of interest, to try to learn more about the treatment. So the question we asked was, how could we do that? And this is particularly relevant in an early phase setting. So, so while the primary analysis may focus on a summary of the digital measure, like a single summary like SB95, and the high frequency is naturally a time series, and the way to get additional answers from this time series would probably imply not summarizing it into just one single measure and using analysis that make use of the full volume of data that is given by this. And this is not going to be straightforward. So as I say, in the SB95 example and in the MBPA, that's an example that will come up a bit later, the strategy that has been taken to treat this is to try to go from the high density measure to one single summary measure and then go back to the analysis that we know. But of course, if we're doing this, if we're condensing, say, 180 hours worth of data in the SB95 into a single numerical value, it's likely that we're going to be missing and not capturing some of the things. So just as much as we all statisticians know that dichotomizing a continuous variable is going to have some information loss, similarly, something like that would be lost in this setting. So the underlying idea is that, so if we want to learn more from this type of data, then one would have to be open to using data that exploits the granularity rather than reduces the granularity. And this is a point that was really nicely made in a paper by Lisi and Avlian, which is where the picture that's shown in the slide now, they were trying to make. So they were also using actigraphy data, and they were just suggesting that perhaps instead of trying to find a single measure, we should put efforts on focusing how the digital measure can augment the information that we have from the traditional or the established clinical outcome, instead of thinking of completely substituting it like SB95 is aiming to do. And so how they proposed to do this was to use the entire time series data and analyze it during a generalized additive model, or a GAM. The GAM is a semi-parametric model that would allow the inclusion of a parametric part and a non-parametric part in the linear predictor. And this non-parametric part could be a spline. It would be smoothing the process. And in this sense, this can be used, this kind of advanced modeling techniques can be used to better understand changes over time in the longitudinal time series, as well as comparing treatment groups. So the picture that you can see here in the slide, it's showing data from 25 ALS patients that they recruited to wear a wearable device to record their physical activity. These patients were followed up at 48 weeks. They were also measured at baseline. They had 12 visits planned at intervals of four weeks. And they were wearing the device for a 24-hour period with a 2-hour period to recharge the sensor. And when they analyzed these data with the generalized additive model, what they produced were smooth curves. In this case, these curves have also a 95 confidence band, both for the baseline, which is the pink or reddish curve, and the blue one, which is the 48 follow-up window. So what you can see from this analysis is that at baseline, there's somehow a clear peak of activity around the hour of the day, the central hours of the day, around 12 or 14. And when these patients are followed up at week 48, this peak is clearly not there. It's already, the curve has shaped and the maximum activity has decreased. So the treatment of the data produced by the accelerometer data, in this case, analyzed in this way, can allow us to identify signs of deterioration, or even, in this case, this was alluded to treatment deterioration, but it could equally be used to understand signs of treatment working at the same time. So this is a possibility, and another example of the same thing comes from our own work. I work a lot with patients with pulmonary arterial hypertension, and we have an example of data coming from an implantable device that is measuring the activity of patients, monitoring it continually and generating this type of high dense data. And the idea is that we could also study through this mechanism of action of treatments, because that could help us understand not only if a treatment works, but how long it takes for a treatment to work. And most of the treatments in pulmonary hypertension work primarily by addressing the symptoms, and the time to address the symptoms by vasorelaxation is a longer time frame. So understanding how long really a treatment can take to learn could have lots of implications on how we design the trial. And one could think that another use of this extra data would be, for example, to help us define futility rules, to understand at which point a treatment could be let go. And the kind of techniques that we're thinking about for doing this are things like time series techniques to detect change point in a series, and to understand, has the treatment work, has something changed at that point. And these are techniques that are not often used in analyzing a clinical outcome measure when it's of the traditional nature. Okay, I think I've said this. So the point here is that in order to answer question 5, we do need to have more flexible modeling approaches that use the full granularity of the data instead of reducing it to one measure. And this will unnecessarily take us to time series analysis and other kind of modeling techniques that might be more complex. But this is the way we think we might get answers to make phase 2 trials most use of this data. So now so as I say, we put it as a question 5, but I think this question 5 can be unpacked as many sub-questions that come within it. But the bottom line for this one is that in early phase, I think instead of trying to select one single summary metric, one really needs to think about how to extract the most information from the high dense time series that we have at hand from the digital device, and try to pair these with the traditional outcome to better answer specific questions. And this will inevitably require statistical techniques that are less often used in clinical trials, and possibly they will require associated extensions of these methods to do that. So before moving into the confirmatory questions of the paper, I want to introduce briefly another example, which is the Bellerophon example. And this example is an example of a phase 2 study in 2019 that evaluated whether inhaled nitric oxide improves physical activity in patients with pulmonary hypertension associated with interstitial lung disease. In this phase 2 study, the outcomes were exploratory, and they were the traditional outcomes in this condition. They included the 6-minute walk distance and oxygen saturation. These are typical traditional measures in this condition, and they included an additional exploratory endpoint that was of digital nature, and this was time spent in moderate to vigorous physical activity, or MVPA. Now, what happened with this study? Of course, we should also say that we are not statisticians in this trial, so we might not be able to give all the answers around this. But the story on the press release was that the phase 2 failed to find a significant effect on the traditional outcomes, that is 6-minute walk and SpO2, but there was a significant effect in MVPA. And therefore, FDA approved time spent on MVPA for a subsequent phase 3 trial with this digital outcome as a primary outcome. And this was also another seminal moment, just like as for SV95. Now, the authors also went on to say that there was an estimation of a substantial reduction in the sample size because of having moved from the traditional outcome to a digital outcome. Using some of the estimated effect from the phase 2, FDA approved a substantial reduction that was around from 300 patients to the order of 140 patients. So basically halving the sample size, and based on the signal that was captured from the phase 2 on this new endpoint. The trial was stopped early for futility, but that's I think less relevant to what we're trying to discuss in this talk. And we are going to use this example of the Bellerophon study to go through the final questions that we want to bring to you in this webinar. So the next question that we have is what about the appropriate estimates when we are changing from a traditional outcome measure to a digital outcome measure? And so estimates can be used in studies with the intention of better interpreting the treatment effects. And we all know that the addendum to the ICH-E9 recommends their use and describes the framework as part of the study. And usually describing an estimate involves specification of these 5 attributes: the population, the variable, the intercurrent events, the treatment, and the population level summary. So with the Bellerophon example in mind, and thinking of the 6-minute walk test as a traditional endpoint and comparison to MVPA, which is a digital version, one can think that that can affect the population in many ways. And this became apparent in our knowledge exchange event. 6-minute walk could exclude people in remote locations because it's an in-clinic type of outcome measure. So patients have to go to the clinic to have their 6-minute walk test measured. However, MVPA could also be excluding those that are unable or unwilling to use digital devices because of privacy concerns. So the population that we would be looking at could be different when we're moving from one endpoint to another endpoint definition. Similarly, the variability capture in the scientific question could be affected. 6-minute walk could be affected by fatigue of motivation. It's a snapshot, one-off kind of measure. So that will affect the activity measured by 6-minute walk much more possibly than a digital endpoint that is capturing patients' experiences more realistically. On the other hand, MVPA could be affected by seasonal or behavioral factors, and Mia will nicely describe this in the simulation study to illustrate that. In terms of intercurrent events, we all know how important these are already in settings in which we are not using digital outcome measures. But the point here is, I think that the type and the frequency of these could be much different when we're having a digital endpoint like MVPA. Think of the technical issues, for example, a software update that changes how the raw data is converted into epoch type of data and so on. So they would be more frequent and they would be different in nature as to what we could expect with traditional outcome measures. In the Bellerophon example, we don't expect that there's an effect on the treatment that we're looking at of the change of the measure. But of course, the population level summary and how it's derived is much more complex when we're thinking of MVPA than when we're thinking of the 6-minute walk, and things like defining missing data suddenly become much more complex. So this was our way of saying that the estimate, when we're using a digital outcome measure, could require much more thinking, specific thinking than what we have already from our traditional outcome measures. And I think at this point, I hand over to you, Mia, again.
Great, thanks, Sofía. So our next question is how seasonal and behavioral factors affect the design and analysis of trials that use digital outcome measures. When you're measuring people's physical activity in their daily lives through a digital device, you're not only measuring exercise capacity, but also people's preference towards exercise And we know from various studies that this tends to be higher in the summer and lower in the winter. So if you imagine in a trial someone who is recruited in the wintertime and their baseline physical activity is measured in the wintertime, and a few months later their follow-up is in the summer, you can imagine that there could be an increase simply due to the seasonal effect. We'll be thinking about this more closely later through a simulation study. But in general, we wouldn't expect to see bias due to the seasonal effect itself, but there could be an increase in the standard error. And similar to seasonal effects, there are other behavioral effects that you may want to be aware of. So for example, when participants wear a device, that can lead to an observer effect where people may perhaps be more active in the beginning because they have a heightened sense of having this device on. For some digital outcome measures that require using an app or doing some kind of cognitive or some kind of physical test, there may be a learning effect, and you may need to consider whether there's an initial familiarization phase that's a little bit different from other attempts. So these are new considerations when you're using digital outcome measures that you may not have if you're using an in-clinic outcome. Our last question, which goes beyond individual studies, is about whether evidence synthesis is possible. We're seeing that there is a growing number of digital outcome measures being used and vast heterogeneity in them, mostly used as exploratory outcomes. In a recent review, it was found that there were 266 different digital outcome measures being used from 21 diabetes trials. So just the vast sheer volume of new digital outcomes that are being developed. This presents big problems, questions going forward when it comes to doing evidence synthesis. So we just like to highlight these 8 questions that we have posed, methodological questions for using digital outcome measures. What we're going to do now is to focus on 3 of them and to illustrate the potential impact of these considerations in a practical example through this simulation, which is loosely based on the Bellerophon phase 2 study that Sofía already introduced to us. So in the study, participants were recruited, and at baseline, they had to wear the accelerometer for 4 weeks, for 1 month. And after that, they were randomized on a 2-to-1 ratio to either the treatment, which was the inhaled nitric oxide, or to the control. And in this study, there were 44 participants, 30 were on the treatment and 14 on the control. Then after a 3-month period, it was the follow-up, and participants again had to wear the device for 4 weeks. And that was, the measurement period was 4 weeks there. We're going to suppose that we have a simplified analysis of this digital outcome measure, and we're going to denote by YIJ the daily time spent in MBPA, moderate to vigorous physical activity, for individual I on day J at follow-up. And we're going to denote by YI bar the average of these daily time spent in MBPA at follow-up for compliant days. I'll come back to what compliant means in a little bit. And in our simulation, we're going to assume a very simplified analysis where we regress the average time spent in MBPA at follow-up to the average time spent in MBPA at baseline, which we denote by baseline I, and the treatment. And some of the questions that we're going to explore are, well, what if some participants are recruited at baseline in the winter and then have their follow-up in the summer, and there is an increase in their physical activity at follow-up simply due to the seasonality? What's the impact of that on this simple analysis model that we've set up? Secondly, if participants have a higher MBPA just in the first week of their follow-up period, so out of the 4 weeks, just in the first week, if they have a slightly higher MBPA because of this feeling of being observed, what will be the impact? And what happens if the measurement period is 2 weeks instead of 4 weeks? What's the impact of this choice about the measurement period? And finally, what happens if you have days that are missing because people remove the device? And this can be according to different mechanisms. So we're looking at, in the first case, when data are missing completely at random, And in the second case, we're looking at when people remove the device specifically on days when they are less active, so missing not at random. So our analysis is on the scale of the average of the daily time spent in MVPA, but we'll be simulating daily time spent in MVPA and introducing these different changes, such as the seasonal effect or the observer effect. Daily time spent in MVPA is right skewed typically, so we have assumed a log normal distribution for this with mean mu ij and standard deviation 46. And the mean has firstly a component for the baseline time spent in MVPA, which we also assume has a log normal distribution. It has a treatment effect, which we assume is 12.5. These values we have taken to be similar to what was observed in the trial. We also have a random effect for patient I. And now we introduced some additional effects. So if there is a seasonal effect, we're going to be simulating this for some of the participants. Then there will be an added effect if someone was recruited in the wintertime, and potentially an additional effect due to an interaction with the treatment. So we're going to allow the seasonal effect to range between 0 and 10, and the interaction will be a proportion of that main effect. Secondly, if there is an observer effect, there'll be again an additional factor added, delta 4, which ranges between 0 and 10, and potentially an interaction with the treatment, which is again a proportion of the main effect. We're also going to be inducing missingness in the outcomes for some participants. And in the study, days were defined as being compliant or not according to a threshold on wear time. So if someone wore the device for over 600 minutes, this was a compliant day. And out of 4 weeks, participants had to have at least 14 compliant days to be included in the analysis. So we're going to look at 2 different mechanisms for how data could be missing. In the first case, this is missing completely at random. So we're going to assume that our completely random selection of days goes missing, and the probability of being missing is unrelated to any variable observed or unobserved. In the second mechanism, we're looking at data that's missing not at random. So we're going to assume that people remove the device specifically when they are less active. So I'll just note before going to the results that our model for the outcome is quite simplified. So if you wanted to have a more realistic outcome, you may want to consider thinking about the autocorrelation between the days, or thinking about the fact that you will have a 0 inflated distribution for time spent in MVPA. But we're going for something that's simple, but will illustrate some of the consequences that we want to show for the 8 questions. So going into the results, at the top here, we're showing the mean of the treatment effect across 10,000 simulations. At the bottom, we're looking at the standard error of the treatment effect across 10,000 simulations. And you can see that in the absence of any seasonal effect, the mean of the treatment effect is around 12.5. That's what we set in the simulation. And the standard error is around 2.8. Now, as the seasonal effect increases from 0 to 10, if 10% of the participants experience a seasonal effect, you can see that there's no bias. However, there is an increase in the seasonal effect. And as this proportion gets bigger, so 20% is in purple, 50% is in light blue, you see that the impact on the standard error is even larger. And on the right-hand side, we're showing what happens if there is an interaction between the seasonal effect and the treatment effect. You can see very clearly now that we've got a bias here and still an increase in the standard error. So that is the impact of seasonal effect. When it comes to observer effect, the story is quite similar. As the observer effect increases from 0 to 10, in the absence of an interaction with the treatment, you don't see any bias. And because we've simulated all participants to have an observer effect, we don't see an impact on the standard error. However, if it's only a proportion of the participants that have this observer effect, you would see an increase in the standard error. On the right-hand side, we're showing what happens if there is an interaction with treatment. And you can see again that we now have bias. And what we're going to show now is what happens if instead of 4 weeks for the measurement period, what if people are asked only to wear this device for 2 weeks? You can see that the bias becomes increased when you have this observer-treatment interaction. And overall, the standard error is much larger. So previously it was around 2.8, and now it's around 4. So we're seeing here how the measurement period can really impact the standard error of the treatment effect. Finally, we're going to have a look at the impact of missing data. In these plots, you're looking at what happens when the proportion of missing days increases from 0 to about half. And in the different colors, you're looking at the proportion of participants who have missing data. They range from 10% in green, 20% in purple, and 50% in light green. And if your outcomes are missing completely at random, you will see no bias, however, potentially a very large impact on the standard error. And on the right-hand side, we're looking at what happens if specifically days that are less active go missing. In that case, you will have an impact on the bias as well as an increase in the standard error. So just to summarize what we've shown in the simulation study, when do you have increase in standard error while having your treatment effect still being unbiased? Well, this happens when you have seasonal effect, observer effect, or outcomes that are missing completely at random. However, if you have any of the following, you are going to have both bias and an increase in your standard error. So that this is when you have season treatment interaction, if you have observer treatment interaction, and if you have outcomes that are missing, not at random. And these are just some of the issues that we've chosen to look at in the simulation study. There may be many other factors that can cause these kinds of effects. Just a note about some strategies for these issues that are in the literature. For seasonality, it may be good to think about recruiting at appropriate times of the year and adjusting for season in the analysis. There are also randomization procedures such as maximum tolerated imbalance, which can quite effectively try to manage to keep a balance between treatment arms as you go on. And this may be good if you have some kind of seasonal effect. For observer or learner effects, there is some discussion in the literature about removing some kind of familiarization period where people may be trying to learn about how an app works or if there is an observer effect. Finally, for missing data, doing sensitivity analyses to missing at random assumption is really crucial. This is something that is part of my previous work, as well as handling missing data on a more granular level. So instead of defining days as being missing or not using some kind of threshold, you could define missing data more at the epoch level, looking at intervals of time that are missing. So what we've discussed today is that there are some key benefits of using digital outcome measures. This includes measuring things that are more relevant to people's lives, measuring things in their daily contexts, and in ways that may enable decentralization so participants don't have to come to the trial center necessarily. And digital outcome measures may be more sensitive. In that case, this can lead to lower sample sizes. And because we get high frequency data from these digital devices, we may gain new insights, and it can even help to add flexibility to studies, for example, by defining new futility rules. On the other hand, there's lots of questions to think about, methodological issues, and we've summarized them through our 8 questions, and these are just some keywords to highlight the things that we discussed. Finally, our kind of point that we like to make is that there's lots of new developments that are happening. This includes the regulatory approval of SB95C by the European Medicines Agency, and also the approval by FDA of time spent in MVPA for the specific case of the Bellerophon trial. We're also seeing frameworks being developed, such as the V3+ framework, and lots of work being done by different consortia, such as Mobilize-D, Clinical Trials, Transformation Initiative, and Digital Medicine Society. However, there's lots of gaps, especially in the methodological areas, and this is what we want to call to attention. So if any of that is interesting, or if you have anything that you'd like to share with us, please do get in touch. And these are the key references that we discussed. So thank you very much for your attention.
Thank you very much for Mia and Villar for your insightful thoughts on digital endpoints and propose 8 key questions and also focus on some questions in detail. Yes, I think the well-framed questions can significantly influence research trends. And thank you very much. And okay, so any questions from our audience? You can unmute and you also can leave your questions in chat. Okay, I see there is a questions on chat. So of the various validation studies you did, which were requested or required by FDA? The question is from Dick Bateman.
So we did not do any of the validation studies, but maybe Mia is better placed to answer some of these. We were just presenting the framework suggested, so we can't comment on which ones were requested or not. I don't know, Mia, if you want to expand on your knowledge of that.